Good morning, students. Now coming to dihybrid cross. So, the cross between two parents that differ in two characters, such a cross is called dihybrid cross. The cross between two parents that differ in two characters, that is called dihybrid cross. In one hybrid experiment, Mendel crossed one homozygous yellow round plant. Okay. In one hybrid experiment, Mendel conducted dihybrid cross between one homozygous yellow round pea plant crossed with another homozygous green wrinkled pea plant. Green wrinkled pea plant. So here there are two characters. One is color of the seed. Yellow dominates over the green. Color of the seed. You know, this is color of the seed is one of the contrasting character. You know, the 7 pairs of contrasting characters. Seed color, yellow, green. So yellow is dominant over the green. Second character, that is shape of the seed. Round shape is dominates over the wrinkled shape. So here Mendel taken two different characters and Mendel selected two different characters. One is color of the seed, yellow, green. Second character is shape of the seed, round, wrinkled. This is the cross between two parents that differ in two characters. That is called dihybrid cross. The characters, the alleles responsible for this yellow is capital Y. Similarly, the alleles responsible for round, capital R, they are dominant alleles. But what are the recessive alleles? Small y, small r. They are the recessive alleles. So here, two characters are there. Every character consists of a pair of genes. These are capital Y, capital R. They are dominant alleles for yellow, round. Small y, small r. They are the genes, alleles of recessive character, green and wrinkled. Now, what about the plants? What type of gametes are produced from these plants? These first plant it produce gametes. First plant it produce gametes with capital Y, capital R gametes. Okay, only one allele is entered into the gametes. Capital Y from yellow, capital R from round. Y R contained in the gene. Similarly, small y, small r alleles are present in the next gamete. So, these plants they produce gametes. Similarly, as a result of the combination of these two gametes, okay, it leads to the formation of F1 hybrid. What are the alleles present in F1 hybrid? Here, capital Y, it is combinated with capital small y. Capital R, small r. There, the alleles present in F1 generation. This is F1 generation. So, the genotypes present in F1 generation hybrid, capital Y and small y, capital Y, small y, and this is hybrid. Okay, this is hybrid. What are the alleles present in this hybrid? Capital Y, small y, capital R, small r. And what is the phenotype of this F1 generation? All the plants formed in F1 generation, they are yellow round plants. All the plants in F1 generation, they are yellow round because capital A dominates over the small y. So that yellow color is established in F1 generation. Similarly, in this pair of alleles, capital R is dominates over the smaller. So that here round shape is established. So dominant phenotype, that is yellow round, is expressed in F1 generation. All F1 generation plants, they are yellow round. Okay, students, now coming to selfing. Scientists also selfed, Mendel also selfed F1 hybrids to obtain F2 progeny, and also explain the combination of these gametes, and also explain the genotypic and phenotypic combinations and genotypes and phenotypic ratios in F2 progeny. Now, for example, this is one plant, capital A small y, capital R smaller. This is considered as male. Similarly, capital Y, small y, capital R smaller, considered as female. This is hybrid plants. Now these hybrid plants, they produce total 4 types of gametes. Total 4 types of gametes are produced from this F1 hybridization. This male plant, it produces male gametes. Total 4 types of male gametes. One is capital Y, capital R, and capital Y, small r, and small y, capital R, small y, small r. Okay? Capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. Similarly, this plant, it also produces the same type of gametes, capital Y, small y, capital R, small y, capital R, and small y and small r. In this way, total 4 types of gametes are formed. Here, they are parents and they are alleles, and this is F1 generation, and this is selfing, and these are also the gametes. Okay, in this way, total 4 types of gametes are produced by the heterozygous F1 hybrid. Okay, now coming to the points. By using this finite square board, we can explain the combination of all these gametes. As a result of the combination of all these gametes, it leads to the formation of 4 into 4. 4 types of gametes are produced from male plant and 4 types from female plant. 4 into 4, total 16 offsprings are formed in F2 progeny. And what about the phenotypes and genotypes of all these 16 plants in F2 progeny? To explain these phenotypes and genotypes, we can use Punnett square board to explain the combinations. For example, this is the Punnett square board. Okay, this is the Punnett square board. Here, total 16 plants are formed. Okay, this is the left column for female gametes, and this is right column for male gametes, and this is female gametes. Total 16 plants are formed. Okay, in the total 16 squares, each square is represented by one combination. Here, total 16 plants are formed in a F2 progeny. Now coming to the male gametes can be taken on the top row, that is capital Y, capital R gamete, capital Y, small r gamete, small y, capital R gamete, small y, small r gamete. Similarly, these female gametes can be taken in left row, capital Y, capital R gamete, capital Y, small r gamete, small y, capital R gamete, small y, small r gametes. They are the female gametes. And combine them and find out the phenotypes and genotypes. First combination, capital Y, capital Y, capital R, capital Y, capital R, small r. Capital Y, small y, capital R, small r. Capital Y, small y, capital R, small r. This is the genotypes of the first row combination. And what about the phenotypes of these first four plants? This is a yellow round. 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 All these four plants, they are phenotypically similar, that is yellow round. But what about the genotypes and phenotypes of the second column? Here, capital Y, capital R, smaller. Capital Y, smaller, smaller. Capital Y, small y, capital R, smaller. And capital Y, small y, smaller, smaller. Okay? And what about the phenotypes of the second row? What are phenotypes? Phenotypes. This is a yellow round. This is a yellow wrinkled. Because smaller is a wrinkled iris, and yellow round and yellow wrinkled. Now what about the third column, and what are genotypes and phenotypes of third column? Capital Y, small y, capital R, and capital Y, small y, capital R, smaller, and small y, capital R, and small y, capital R, smaller. Okay? What are the phenotypes of these plants? This is a yellow round, and also yellow round, and this is a green round. And what about the next one? This is also green and round. Okay? Yellow round, green round. Okay. Now coming to another one. This is the combination capital Y, small y, capital R, smaller. And capital Y, small y, smaller, smaller. And small y, capital R, smaller. And small y, smaller, smaller. Okay? What about the phenotypes of these four plants? Yellow round and yellow wrinkled. Okay? And green round. And this is a green wrinkled. In this way, total 16 plants are formed in F2 generation. This is F2 generation. This is F2 generation. Okay, students, what about the phenotypic ratio and genotypic ratio of this F2 progeny of dihybrid cross? To explain the genotypic and phenotypic ratios, we can use another table. Now coming to another table that explains the genotypic and phenotypic ratio of F2 progeny in dihybrid cross. Now this is the second table. Okay, this second table is very important. Okay, this is the second table. Okay. This is genotypes. Okay, genotypes, and this is genotypic ratio, and this is phenotypic ratio. This is genotypic ratio. Okay. Now capital Y, capital R. How many capital Y and capital R alleles are there? How many capital Y, capital R alleles are there in this table? Only one is there. Only one. Okay? Only one. But how many caplar small are there? Caplar small are. This is two plants are there. What are these two plants? What are the two plants? One. This is one, and this is second one. So caplar smaller. There are two. That is two. Okay, but how many caplar genotypes are there? Caplar genotypes are there. That is only one. Only one. Okay. This is one, and next one. This is the next one. There are two. That is two. Okay. And how many capital Y small y caplar smaller are there? Capital Y small y caplar smaller are there? There are total four. 1, 2, 3, 4. So all these plants, they are yellow round. All these plants yellow round. Total 9 yellow round plants are there. 12, 24 means this is one, and this genotypes 2, this genotypes 2, this genotypes 4. Total 9 yellow round plants are there. Okay, total 4 yellow round plants are there. Okay, now coming to another one. How many capital Y smaller genotypes are there? That is only one is there. And where is location? This one. Okay, only one is there. This type of genotypes, they are present only one. Okay, only one, and this one. And how many these combinations are there? Capital Y small y, capital Y small y or smaller. That is 2. There are 2. 1. Okay, and second. There are 2. And these plants are yellow wrinkled. Okay, they are yellow wrinkled. How many yellow wrinkled plants are there in this table? That is total 3 plants. Okay, and how many green rounds are there? Green round means small y, caplar, small y, caplar, 1. Okay, only one is there. This is 1. And how many small y, caplar smaller are there? How many small y, 1. And this is 1. And next one. Okay, this is 1, and this is one genotype. Okay, and another genotype. Find out the next genotype. This one. Okay, so there are 2 plants. These 2. Okay, these 2. There 2, and these plants are the green round. Okay, green round. How many green round plants are there in this list? Total 3. And how many genotypes are there? Small y, smaller. Only one genotype is there. This is 1. This is green wrinkled. This is green wrinkled. Only one plant is there. Okay. So this is the genotype ratio, and this is phenotypic ratio. What is the phenotypic ratio? What is the phenotypic ratio? What is the phenotypic ratio of dihybrid cross in F2? That is 9 plants are yellow round, 3 plants are yellow wrinkled, and 3 plants are green round, and only one plant is green wrinkled. So 9:3:1. This is the phenotypic ratio of dihybrid cross F2 proceeding. But what about genotypic ratio? Genotypic is this one is the genotypic ratio. Genotypic ratio of yellow round 1:2:2:4. Genotypic ratio of yellow wrinkled 1:2. Genotypes of green round 1:2 and green wrinkled 1. That is 12, 24, 12, 12, 1. This is genotypic ratio of F2 proceeding. So phenotypic ratio is 9:3:3:1 and genotypic ratio 1:3:2:4:1:2:1:2:1. Simply this is and expressed as 12, 24, 12, 12, 1. This is genotypic ratio. Students, here one of the most important point, that is there are total 4 phenotypes. One is yellow round, second one is yellow wrinkled, third one is green round, fourth one is green wrinkled. These first phenotype and last phenotype, they are parental phenotypes. Yellow round parental phenotype, green wrinkled parental phenotype. But what about these two? Yellow wrinkled and green round phenotypes, they are additionally formed in F2 progeny. In addition with the 2 phenotypes of the parents, 2 additional phenotypes, they are called recombinant phenotypes. In addition with the 2 parental phenotypes, yellow round and green wrinkled, 2 recombinant phenotypes called yellow wrinkled and green round phenotypes are formed. Total 4 phenotypes are formed. Of these, 2 phenotypes are parental phenotypes and remaining 2 phenotypes are recombinant phenotypes. Okay, so there are total 4 phenotypes. This is the phenotypic ratio and this is the genotypic ratio.